Assalamualaikum. My name is FARIHAH binti ADNAN. I am from AS246B and today I will be presenting my research entitled T-cell epitope prediction of dengue virus Denv, non-structural-1 NS1 gene using Tepitool prediction tools. This study focuses on dengue virus, which is a major public health concern in Southeast Asia. Dengue has 4 distinct serotypes transmitted by Aedes mosquitoes. Current vaccine strategies have limitations in terms of the ability to provide effective protection against all 4 dengue serotypes. For example, traditional vaccine strategies have shown the ability to provoke immune responses but fail to provide protection across serotypes and are also time-consuming. Therefore, this study applies the reverse vaccinology and immunoinformatics approach to identify potential T-cell epitopes from the dengue virus NS1 gene to contribute to the development of a multi-epitope dengue vaccine. The strategy is faster, cost-effective, and enables the identification of conserved and specific epitopes across diverse viral strains. This study was designed with 3 main objectives. First, to retrieve the non-structural-1 or NS1 protein sequences of dengue virus from all 4 serotypes using the NCBI virus and GenBank databases. This ensures that the analysis is based on reliable and regionally relevant sequence data from Southeast Asian countries including Malaysia, the Philippines, Thailand, Indonesia, Vietnam, and Myanmar. Second, to predict epitopes from using the Tepitool database. This objective aligns with the need for a safer and more targeted vaccine approach by identifying peptides with strong binding affinity to MHC classes. Finally, to determine the most conserved regions among the predicted T-cell epitopes across all 4 dengue serotypes, because highly conserved regions increase the potential for cross-serotype protection. Next, the methods in this study were designed to address each objective. First, NS1 protein sequence from all 4 dengue virus serotypes were retrieved from the NCBI virus and GenBank databases using sequences from Southeast Asian countries. This step supports the objective of obtaining reliable and regionally relevant sequence data. Next, T-cell epitope prediction was carried out using Tepitool to identify potential epitopes from the NS1 protein. Peptides were selected based on their percentile rank, which reflects binding strength to MHC molecule. Next, the epitope conservation analysis was performed using the IEDB database to evaluate whether each predicted epitope was conserved across different dengue virus sequences. Then, it was visualized using web logo, where highly conserved amino acid positions appear as the tallest stacks. Following this, epitope similarity analysis was conducted using the BLAST-P to assess similarity between the predicted epitopes and human proteins. Next, this slide summarizes the data set used in this study. A total of 48 NS1 protein sequences were retrieved from Denv1 to Denv4 using the databases. The NS1 regions were extracted from dengue polyprotein sequences to ensure accurate analysis. Samples were selected from Southeast Asian countries including Malaysia, the Philippines, Vietnam, Thailand, Indonesia, and Myanmar due to high dengue endemicity. Using multiple sequences from different countries and serotypes helps reduce bias and ensures representative genetic diversity for downstream analysis. Next, this slide shows the T-cell epitope prediction results for the NS1 protein. Predictions were performed using Tepitool, which identifies peptides that bind to 12 MHC class 1 and 6 MHC class 2 alleles. Epitope selection was based on percentile rank, where lower values indicate stronger binding. For class 1, below 1, and for class 2, below 10. Several strong binding epitopes were identified, and some appeared in sequences from multiple countries. For example, FQADSPKRL was found in both Philippine and Vietnamese sequences, suggesting possible conserved immunogenic regions. This slide shows the conservancy analysis of the predicted T-cell epitopes from the NS1 protein. A total of 6 conserved epitopes were identified from MSC Class 1 and 2 MSC Class 2 epitopes. Only epitopes with 80 to 100% sequence conservation across all dengue serotypes were selected to ensure broad applicability. The conservation levels were assessed by comparing sequences from Denv1 to Denv4, and the conserved regions were visualized using Web Logo 3.0 as shown on the slide. Next, this slide compares our predicted conserved epitopes with previous study. The epitope HGWTTEQYKF was previously reported as a T-cell epitope, which supports our prediction. Another similar epitope, TWTEQYKFQ, was also identified in earlier NS1 studies. Next, the blast-based analysis was performed as shown in the figure. Most protein product from the 4 dengue serotypes was screened against the human proteome. The results confirmed that these sequences were ideal properties of a potential vaccine candidate that targets the pathogen specifically without triggering an accidental autoimmune reaction in the host. In conclusion, this study demonstrates that bioinformatics and immunoinformatics tools provide a safe, rapid, and effective framework for early-stage vaccine design. Using the Tepitool database, 8 T-cell epitopes with strong binding affinity were successfully predicted. These epitopes were highly conserved across dengue virus serotypes, indicating their potential to induce cross-serotype T-cell immune response, which is essential for effective dengue vaccine development. For future work, further in silico screening such as antigenicity, allergenicity, and toxicity analysis is recommended to strengthen epitope selection. In addition, evaluating population coverage across broader ethnic groups and incorporating B-cell epitope predictions to enhance the overall immune response and support the development of a more comprehensive multi-epitope vaccine.